Activation of NLRP3. NLRP3 is a cytosolic receptor in your innate immune system. There is a long list of things that have been shown to activate NLRP3. We have a heap of different types of receptors in our bodies, and most of the time when they get activated, it's because something binds directly to them. The thing that binds we call a ligand. So insulin is the ligand for the insulin receptor. And like the insulin example, the ligand is usually pretty specific to the receptor. Insulin binds an insulin receptor, but it won't bind an adrenaline receptor. But no directly binding single ligand has ever been found for NLRP3. Just a whole bunch of things that seem to activate it, but we don't quite know how. Many of the known activators seem to converge on a common theme of potassium efflux, which just means potassium is leaving the cell. Healthy cells contain a set amount of potassium, so when too much potassium exits the cell, it's a sign that something's wrong with the cell, and NLRP3 will kick off. While we don't know exactly how it happens, we just know that it does. Most NLRP3 research talks about NLRP3 activation occurring in two stages. The first stage is called priming, and the second stage is called activation. Priming happens after a first signal. In the lab, they trigger this priming step by applying LPS, or lipopolysaccharide, which is also known as endotoxin. It comes from bacterial membranes, so it's not really something that you'd want in your cell, and it gets the attention of NLRP3. In the first signal or priming step, the NLRP3 protein changes its conformation or shape to become primed and ready for activation. The other thing that happens in priming is that more NLRP3 proteins are made, and more of the other parts of the NLRP3 inflammasome are made too, so the cell stocks up on inflammasome components to ready itself. But to get to the next step, the activation step, it needs a second signal, and that could be the potassium efflux thing, or maybe some reactive oxygen species which are bad and come from damaged mitochondria in our cells. In the lab, the second signal applied to cells is often nigericin, which is great at inducing potassium efflux and triggering full NLRP3 activation. So what's the deal with CAPS and NLRP3 activation? With the NLRP3 mutations that cause CAPS, we know NLRP3 is hyperactive. It is thought that our NLRP3 proteins are always primed, so we don't need two signals. In 2020, a paper came out describing computational modelling that was done with 177 different CAPS mutations, covering the spectrum of severity in the CAPS syndromes. Their findings concluded with two different scenarios. One was consistent with the idea that CAPS mutant NLRP3 proteins take a primed conformation or shape. The other scenario was that mutations affect how NLRP3 interacts with other proteins. Both scenarios support the idea that CAPS mutant NLRP3 proteins need only one signal to be activated, not two, and how it happens just depends on where your mutation is. But what about the other aspects of priming? The cells loading themselves up on NLRP3 and inflammasome components. Is there a point where we CAPS mutants hit a critical mass and don't even need an activation signal? NLRP3 inflammasomes are formed stoichiometrically. That's just a really fancy way to say that if there are enough primed NLRP3 proteins around, they will do the inflammasome thing spontaneously. There are certainly triggers with CAPS that cause really bad flares, but before I was diagnosed, there was never a day that I wasn't symptomatic in some way. It was just a matter of scale. In a 2018 paper, researchers found that CAPS monocytes had high basal levels of reactive oxygen species, confirming some of their earlier work. Monocytes are a type of white blood cell, and high basal reactive oxygen species means these cells are showing signs of stress without any stimulation. Using these cells in a dish, normal monocytes needed two stimulation signals to activate NLRP3, but the CAPS monocytes only needed one. The cells that they used had these mutations, which are all in or adjacent to the NACH domain of NLRP3, supporting the idea that mutations in this region of NLRP3 result in an always primed state. As a side note, it will be interesting to see if this is also true for non-NACH domain mutations, like Y859H, for example, if there are any researchers watching. With all of the new NLRP3 inhibitor drugs in the pipeline, we just hope they'll be able to close up our mutant NLRP3 into an inactive state. That would be super great.